Good afternoon, everyone. Let me rather congratulate Joseph and his team for conducting a wonderful program, and also thank him for inviting me to chair this session. Post-lunch session, the first topic is on biosimilar insulin. Yeah, just a word about biosimilar insulin. It's actually a copy of the original molecule, biological molecule, and the key question is whether the relevance of this small change will have any relevance to the clinical practice. To address on this topic, I would like to invite Dr. Mathew John for the talk. And about Mathew John, he completed MD General Medicine from Christian Medical College, Vellore, and DM from KM Hospital, Mumbai. He has more than 30 mixed publications and chapter in textbooks and more than 25 conference publications. Right now, he's a consultant endocrinologist at Providence Endocrine and Diabetes Specialty Center, Trivandrum. Over to you, Dr. Mathew.
Thank you. Okay, thank you, Jyoti, for this wonderful stage. Really pleased to see the number of people attending the meeting. Okay, I'll just move on to the topic. I think we are running late. Biosimilar insulins. So to take you through this topic, I'll be trying to answer certain questions and then finally arrive at sort of conclusions which Dr. Radhakrishnan just told. So I think this is one of those topics which is not very commonly taken up in meetings. So I'll just go through the basics and I'll probably sum up with what a biosimilar insulin is. So biological is something which we need to understand first before we understand biosimilars. No problem. Okay, thank you. So biologicals are therapeutic biologicals or biologics. These are all different words for a biological. So basically biologicals are substances which are medicinal substances which are generated by living organisms, cells or tissues. And they are usually made by recombinant DNA techniques. So this is a sort of theory behind biological. So if you look at examples of biological, then you become more familiar with this, like insulin, growth hormones, erythropoietin, gonadotropins, GLP-1 receptor agonists. These are all biologicals. So what is it? What is the other side of it? The other side is chemical molecules. Chemical molecules like saxagliptin, Saxa has not paid me for sure, aspirin, glycoside, these are all molecules which are chemical molecules. So biologicals on one side, chemical molecules on the other. And the difference is this, the biologicals are significantly larger in size. So this is a diagram which shows aspirin, insulin, erythropoietin and a monoclonal antibody. And what you can see is the aspirin is such a small molecule compared to biologicals like erythropoietin. So the difference is biologicals are more complex molecules and it's much more difficult to make them. So what is a generic versus a biological? So what, you know that just like Dr. Shashi Tharoor told, once the patent on the innovator molecule expires, then you have similar molecules being brought out by the different pharmaceutical companies. So for a chemical entity, if a similar molecule is brought out, it's a generic. But for a biological, if a similar molecule is brought out, it's a biosimilar. So now you understand what is the difference between a generic and a biosimilar. And you already knew how a biologic differs from a chemical molecule in terms of the structural complexity. Now, how does the regulatory authorities define a biosimilar? So biosimilar, by the word similar itself means it is similar to the innovator molecule in terms of certain characteristics, which includes the biological activity, the safety, and the quality characteristics of the molecule. So this is a biosimilar, and it is similar to the original innovator molecule. That's what a biosimilar basically means. So why is this big confusion about a chemical molecule versus a protein molecule? So what you can see on your right side are two chemical molecules. One is sitagliptin and the other one is glimepiride. And you can see the simplicity of these molecules when you see the chemical structure of them. But on your left side, you have a protein structure. And if you go back to your biochemistry, you would realize the protein is not at all, it's not just a combination of amino acids. The combination of amino acids is only the primary structure of the protein. And beyond the primary structure, the protein has got a secondary structure, a tertiary structure, and when multiple things join together, there's a quaternary structure. And there are additional things which after a protein gets synthesized, you have post-translational modification of the proteins. So compared to a chemical molecule, a protein molecule is entirely different in terms of the structure, and that's the difference between a generic and a biological, which actually makes it. Now, how are these molecules developed? So if you go back to the drug development for an innovator molecule, it's actually a very painful thing. If you look at this thing, the first part is the drug discovery. When you discover a drug, you realize the mechanism of action of the drug and how that drug can help a patient control his diabetes. So out of the 50 to 5,000 to 10,000 compounds which gets discovered, finally only one FDA approved drug comes out of it. So after the drug discovery, you have preclinical studies with the drug. And after the preclinical studies, then you have the clinical studies with the drug. And then the drug goes for a new drug approval, and then the drugs gets approved for clinical use. So I'm going to concentrate on the preclinical drug development area alone from the aspect of biosimilars. So if you look at the preclinical drug development area, that itself is quite complex. And it involves formulation studies, it involves safety studies, it involves animal studies, toxicology studies, pharmacokinetic, pharmacodynamic studies. And after all the studies are completed, you have the preclinical data, and then you move on to the clinical data. So this is the preclinical state, and then you go on to the clinical side. I'm not going any further on that, but I'm now trying to compare the regulatory process of how a biosimilar gets approved compared to that of how a originator gets approved. I showed you the originator method, how it goes through the initial discovery phase, the preclinical phase, and the clinical phase. And now if you look at the biosimilar, when a biosimilar is made, already the early stages of development is known. What is the molecule? What is the chemical structure of the molecule? What is the toxicity of the molecule? All these things are known. So the biosimilar person does not need to go through all those initial part. But after a molecule is made, the biosimilar development program has to convincingly show that the biosimilar molecule which is developed is similar to that of the original molecule, especially in terms of the constitution of the molecule, and then also in terms of the PK/PD, the pharmacokinetic, pharmacodynamic properties of the molecule. And finally, if all these things match, you know that this is very similar to the original molecule in terms of the action. So mainly the biosimilar development program is different from the originator program in terms of more of PK/PD studies, I'll again come to this data later. But even the initial studies of these molecules are not very simple. You can see the amount of data a company has to generate before a biosimilar comes in. It's not copying a generic chemical molecule. A biological has to go through a lot of areas before it can just go in for a new drug approval. And again, a different in vitro studies to show that this is very similar to the originator molecule in terms of the various different parameters in the preclinical stage. Now coming to two words, I think these are words which we learned in pharmacology. One is pharmacokinetics, that is what the body does to the drug in terms of absorption of the drug, distribution of the drug, metabolism, and finally the excretion of the drugs. This is the pharmacokinetic, which we call as the PK. And then we have the pharmacodynamics of the drug, what the drug does to the body. In terms of, example, if you are using an insulin, how much of sugar can the insulin reduce? That is the pharmacodynamics of the molecule. Now, all these regulatory bodies insist that the pharmacodynamics and the pharmacokinetics of this biological or the biosimilar that you develop matches very closely with that of the biological or the innovator molecule. So they have given a margin of safety, and only if the biosimilar satisfies this margin of safety, then can the biosimilar get an approval. And different organizations, the FDA, the European Medicine Agency, the Japanese PMDA, they have got different regulations on how a biosimilar can be approved. And these are the different regulations. I'm not going into any of those details. Now, biosimilars have been in market for some time. And these are different biosimilars which we all use in our practice, either in India or throughout the world. Growth hormone is a biosimilar. It's almost growth hormone. We don't say it's a biosimilar anymore because we have been using different brands of growth hormones and different types. Erythropoietin alpha, insulin glargine is the one we are speaking about. Infliximab, folitropin alpha, factor VIII, these are all biologicals or biosimilars which are already there in the market. Now coming to the last part of the talk, that is the insulin glargine biosimilar which we are concerned about. Lantus is the original product of Sanofi. The patent expired in February 2015. And since then, glargine being a very hot molecule in the market, multiple pharmaceutical companies are in the process of developing biosimilars to glargine. Basilog by Biocon and Abesaglar by Eli Lilly are approved biosimilars of glargine. And Basilog has been approved in 25 countries, including Japan. Japan comes up, including Japan means Japan is one of the most stringent markets to get a drug approved. And getting approval in Japan itself is an achievement. That's why we put including Japan. And if you look at a drug development program of, or just the manufacturing of a molecule like glargine, you would realize how complex it is for the molecule to get isolated, to have a vector, to put it into the host cell, then the folding, the enzymatic cleavage, the purification, and the different processes before a molecule can get into the market. And if you look at the clinical development program of Basilog, it has gone through different countries, and you can see the different countries where this molecule has been approved or is in the process of getting approved. I'm not going into this complexity of doing a PK/PD study, but just to summarize what a PK/PD study, how the final results of the PK/PD studies looks like, it is something like this. You can see two lines which are going very closely together. This is actually the pharmacokinetic comparison of glargine, the innovator molecule, to that of the biosimilar. When you give a single dose of a molecule, glargine, and you look at how the glargine concentration behaves in the human body across a time period, and that this is how the graph looks like. And you can see the biosimilar almost exactly following the curve of the originator molecule. And if you look at the pharmacokinetics, you can again see that this is the glucose lowering. That means this is something called a clamp study where you have the glucose lowering effect. Again, you can see this is almost matching that of the originator molecule, showing that this is very good biosimilarity between these two molecules. And this, for clinicians, we are not much happy about the PK/PD studies because for us, molecule should reduce the HbA1c or reduce the blood sugar. And that is actually what matters to us as clinicians. And this difference in A1c between the originator molecule and the comparator molecule in terms of A1c lowering, almost the same. So be it a PK/PD study or be it an A1c lowering, the efficacy studies, and including safety studies, these two molecules behave very similarly. So what is this rationale for using biosimilars? Why can't we just persist with the originator molecule? Why do we really need biosimilars? The most important thing about biosimilar is the affordable drug cost. Whenever a biosimilar comes into the market, Sarcio is actually the filgrastim which came to the market. There is a discount in prices because the biosimilar development program is not, the clinical part of the biosimilar development program is much less than that of the innovator molecule. And so the amount of money spent in the clinical trials of these molecules are much less. And so the biosimilar producing companies can obviously sell the molecules at a cheaper rate compared to that of the originator molecules. Second, once these molecules, everyone would like to use a good molecule. And when the price of the molecule comes down, naturally, more patients will have access to the molecule. And so the number of patients using the molecule in the community will go up, and more patients will get the benefit of using the molecule. And again, biosimilar technologies are more recent compared to the innovator molecule technologies. I'm not discounting the fact that even the innovator technologies keep changing. But you are getting more exposure to the modern technologies. So just to summarize this thing, the innovator molecule goes through a biological process of manufacturing it. Then you prove the structure, the mechanism of action of the molecule. Then you go for a PK/PD studies. You prove the safety of the molecule. And then you go into an extended efficacy phase, the clinical research. But when you come to a biosimilar, you can see proving the structure, safety, biological is very important. But then you go, major thing is that you are trying to show that this molecule is similar to the original molecule. And your efficacy or the clinical trial part of it is actually very short. And that is exactly the place where the molecule saves the money, which gets translated to a more affordable molecule in practice. So the main exercise of a biosimilar development program is to provide evidence of similarity, that it is similar to the original molecules. And the regulators look at a totality of evidence, not at one specific or two specific areas. But again, the next question which comes is that when glimepiride, the initial glimepiride was priced at 15 or 20 rupees, the generic glimepiride is priced at less than 1 rupee. Then why is it that biologicals, even though they are cheaper to produce, are not heavily discounted? This because of the same fact that I told you, a biological is not produced just like that in a reaction. A biological is a very difficult task to produce, which has go through the different cycles. So even to manufacture a biological, it's not a very cheap method to do it, and that is the reason why the price of biologicals are less than that of the innovator molecules. But still, they are not like damn cheap things. Now, when you think that making a generic seems to be a very easy game. But why is biologicals not an easy game? Because there have been major problems in the past. There are two stories which I would like to say before I end the thing. One is the Marvel insulin story. Marvel Life Science is a Bombay-based company which actually submitted a drug application for insulin, regular insulin, NPH insulin, and a premixed insulin. But even though these two insulins, the comparator and the Marvel insulin had the same primary amino acid structure, the pharmacokinetic and pharmacodynamic properties of this insulin were entirely different from that of the original molecules. And so this approval was denied. Second is the case of an erythropoietin story. This is actually not a biosimilar story. The story is actually the originator molecule itself changed its formulation only to realize there is an increased risk of pure red cell aplasia with the new preparation compared to the original preparation. So again, to fact that even if you change the method of making a biological, there can be a difference in the biological and that can have adverse events. So it is not very easy to make a biological, and it's a difficult game it's not for everyone to play the biological game, even though you think that it's a copy-paste game. And the world over, there is an increased global demand for biosimilars. And I think biosimilars are going to stay. And you can look at examples like major companies like Lilly and all the innovator companies now moving on to biosimilar production, showing that cost cutting so that more patients can afford it and more payers can afford it would be the way forward for biosimilars. I stop here and finally I go through a summary. Biosimilar formulations are similar to the innovator but not the exact replica. They are the same active ingredient and a very similar PK/PD profile and similar efficacy characteristics. They are cost effective. The regulatory approval process of biosimilars are not very easy. It's quite stringent with the FDA or the EMA or the Japanese authority. It's quite stringent. And the approval of a biosimilar glargine by Biocon in the Japanese PMDA, it's actually a proof of the high quality biological manufacturing capacities of India and the process and the standards the company is able to follow. I leave you here. Thank you very much.
Thank you, Dr. Mathew John, for that slightly difficult talk to a simple one. And I think the session is open for discussion now.
I forgot the question. Now, what about the long-term carcinogenesis? You didn't tell us about the pharmacogenetics. And one more thing which I forgot.
Okay. Yeah, it's okay, sir. See, what you do is that I showed you that different preclinical data which is generated. And one of the...
Cardiovascular safety.
Okay. I showed you about the preclinical data which is generated. And one of the components of the preclinical data is to look at the insulin growth factor 1, actually the affinity of the insulin to the insulin growth factor 1. That is one of the preclinical requirements of insulin generation data. I showed you a lot of preclinical data. I didn't go through individual things. So basically this data is the one which looks at the cancer potential of these drugs. But you won't have long-term safety data when a biosimilar comes into the market. For that purpose, actually for both biosimilars and for the original molecule, there is a very strong pharmacovigilance program which runs to identify whether all these things are there. The long-term risk of carcinogenicity, the long-term risk of immunogenicity, the long-term cardiovascular risk, all these things are being generated in the post-marketing trial. It is not done during the clinical development process. So when you get an approval by FDA or by the European agency or by the Japanese PMDA, the company has to submit a very strong pharmacovigilance or the risk management program on how they are going to look at these aspects in the post-marketing stage. You won't get that in the approval stage. It's not required in the approval stage. But it is believed that most of these are going to be similar to the originator molecule. Rajendra.
Yeah. Dr. Mathew, what is your personal opinion regarding the patent right of holding a patent right of a products like natural biologics, as there is no discovery on it? It is only the process of isolation or identification. How beyond the concepts of industry you justify the patency of a biological compound of natural occurrence?
See, I think biosimilars don't have any patent because any number of biosimilar
Not
development
the
is
biosimilar, for
biological.
example, growth hormone is not a discovery, it's a natural compound. Somebody has isolated.
I understand.
It
Yeah.
is only the industry which has the process of isolation, but in the patent law as the clause,
It is
there
there.
should be a discovery or a addition of something like a physiological action. But what is growth hormone isolated is same as the growth hormone in our body. Then how you justify the patent of growth hormone for an industry?
I think I won't comment on it because personally I don't have any sort of that sort of thing. But actually what you told is true. But I think even if it was such an easy job for everyone to do, I think most people would have done it. But finally, if you look at production of insulin, like the production method is proprietary to the company. See, if you look at the growth hormone or an insulin, example, if you go back in history to how Lilly produced insulin or how Novo produced insulin, each company has got a proprietary method of producing insulin. Like Biocon uses Pichia for their process. Sanofi uses an E. coli for their process. Other one uses a Saccharomyces yeast for their process. So even though it is a natural or a biological compound, the method of production is actually proprietary to the company, and the company has spent or has got the knowledge base to develop that product. So I think it should be granted, but I'm not going into the ethical or legal aspects of that. I'm very sorry that I'm not.
Biosimilar action like Clemiprid generics cost will be explained once again, sir.
I didn't get you. Can you please repeat that question?
Biosimilar actions like Clemiprid
Yeah.
tablets, it is a cost of the price will be reduced in generic items or not?
You're asking me whether the price of
Cost.
will be less. Yes, that's what I told you. See, you
Once
cannot-
again, please repeat.
Yeah. Actually, developing a biological is almost like developing a new molecule. The only thing is that you know the molecular structure of this molecule. Like glargine, you know this is how the molecular structure looks like. But you are going through an entire process of developing it. Like, example, the glargine, original glargine is developed in the E. coli. But the new glargine by Biocon is developed in a Pichia. It's another organism. But then this entire process is, only thing is you know that molecule is there. But to replicate that process and go through the purification and the crystallography and all those things itself is a big process. Unlike a molecule like sitagliptin or saxagliptin getting reproduced by a chemical reaction. And that is exactly the reason why they are not going to be very cheap. They are going to be cheap, but they are not going to be like very low priced.